Hi, good day everyone. My name is Muhammad Mukir Iskandar bin Muhammad Norazwan. Today I will introduce you about morphology, anatomy, phenology and photosynthesis of maize. And for the course of IGL 499 under the supervision of Dr. San Fuying. Maize is one of the most important cereal crops in the world and is widely used for food, livestock feed and industrial purposes. First, morphology. Let us discuss the morphology of maize. Morphology refers to the external structure of the plant. Maize has a fibrous root system that helps absorb water and nutrients while providing support. The stem, also known as the stalk, is erect, cylindrical, and divided into nodes and internodes. It can grow up to several meters in height depending on the variety. The leaves are long and narrow with parallel vines, which is a common characteristic of common plants. Maize is monoecious plant, meaning male and female flowers are found on the same plant. The male flowers from the tassel at the top, while the female flowers develop into ears on the sides of the stem. The fruit of maize is called a kernel or grain, which contains the embryo and store food for germination. Next, the anatomy. What is anatomy of maize? I will explain the anatomy of maize. Anatomy refers to the internal structure of plant organs. In the root, tissues include the epidermis cortex, xylem and phloem. These tissues help absorb and transport water and nutrients. In the stem, vascular bundles containing xylem and phloem are distributed throughout the ground tissue. The leaf contains mesophyll cells and bundle sheath cells surrounding the vascular bundle. This arrangement is called crans anatomy, which is a characteristic feature of C4 plants such as maize. Crans anatomy improve photosynthetic efficiency and helps maize perform well under hot conditions, such as today we have the maize and under the sunlight. So crans anatomy will allow maize to perform photosynthesis very well. Next one, we have photosynthesis. What is photosynthesis? Maize use the C4 photosynthesis pathway. In C4 photosynthesis, carbon dioxide is first fed in the mesophyll cell before being transported to the bundle sheath cell for sugar production. This process reduces photorespiration and allows maize to use sunlight, water and carbon dioxide more efficiently. As a result, maize grows well in high temperature and bright sunlight and generally produce higher yields and compared to many C3 crops. Next, we have phenology. Phenology refers to the growth and development stages of the crop. First stage is germination, where the seed sprout and the seedling emerges from the soil. Next is the vegetative stage, during which leaf, roots and stem develop rapidly. The reproductive stage begins with the tasseling and silking. Pollination occurs when pollen from the tassel reaches the silks of the female flowers. After pollination, kernels develops and fill with nutrients during the grain filling stage. Finally, the crop reaches physiological maturity, where kernels attain maximum dry weight and are ready to harvest. Depends on the variety. Maize usually matures within 100 to 140 days after planting. Now we have the conclusion. In conclusion, maize is an important cereal crop with a fibrous root system, erect stem, and specialized leaf structure known as crans anatomy. Its C4 photosynthetic pathway allows efficient growth under warm conditions, while its growth process progresses from germination to maturity through these distinct development stages. Thank you for your attention.
4-carbon compound. Like C3 plants, C4 plants will close their stomata to conserve water loss. However, unlike C3 plants, C4 plants will continue to make sugars using photosynthesis instead of entering photorespiration. They do this by initially fixing carbon dioxide in the mesophyll cells via the enzyme PEP carboxylase, which reacts the 3-carbon phosphoenol pyruvate, or PEP, with CO2 to form the 4-carbon oxaloacetic acid, or OAA. The resulting 4-carbon compound within the mesophyll then cycles into the bundle sheath cells and releases CO2, meaning the levels of carbon there will always remain high enough for the Calvin cycle to make sugars. It's important to note that the utilization of C4 carbon fixation does not indicate common ancestry. About 3% of land plants use C4 carbon fixation, most of which are monocots. However, C4 carbon fixation has evolved independently at least 20 times in the grass family alone, and is thus an excellent example of convergent evolution.